So it's great to see everyone here. I hope you had a good time last night. And so this is just the opening of our 21st annual NeuroMusic conference. So I'm very excited to welcome everyone here. I wanted to acknowledge, first of all, that McMaster University recognizes and acknowledges that it's located on the traditional territories of the Mississauga and the Haudenosaunee nations, and within the lands protected by the Dish with One Spoon Wampum Agreement. And in addition to that, I just wanted to say that we know that music has been made on these lands for a very long time, and we honor those traditions. And we seek now to understand the importance of music for individuals, communities, and all societies. So welcome. Wi-Fi information in between talks and so on, it will be up on the board, but just so you know, if you have a McMaster Wi-Fi, please use that, or use EduRoam if you have that, and if not, there are a limited number of McMaster Wi-Fi codes available at the registration desk. Also join us on Discord for further conversations, and we ask that you use your name under which you submitted your registration or poster so that people can find the same person from the poster and on Discord. So I'll just leave that for a second if anybody needs to use their phone. Also wanted to say that tonight is going to be amazing. We have a keynote lecture and concert that we have every time at these, and happy to say our guest speaker is Dr. Haley Cragness, who's also giving a talk tomorrow. And the title of our talk tonight will be Musical Meaning in Childhood. We're also really happy to have Versacello. They are sort of, they've played many times at the Live Lab. They're an amazing cello duo. They arrange and compose all kinds of music, so that will be a treat as well. The bad news is that it is sold out, but you can inquire at the registration desk if you didn't get your tickets already, if you want to go on the wait list. And the good news is that the live stream is free, so you can still, even if you don't have a ticket, you can still watch on the live stream. There are so many people to thank. First of all, a big shout out to Elaine Wiskin. Where are you, Elaine? She's probably out in the lobby doing something. I'm sure you all got emails from her. Every year she's the one that makes sure that the conference runs smoothly and that everyone is happy. So if anything, if you have a complaint, go and talk to her. No, you can actually come and talk to me or any of the other people. But just also a call out to Hanny Tofik, Susan Marsh-Rollo, John Everson, Lauren Fink, Dan Bosniak, Ian Bruce, and Cynthia Belaski, who really helped put it together. And then our organizational volunteers who also put in a lot of time, Dan Cameron, Amanda Han, Luke Klein, Sarah Ripley, Rafa Roman Caballero, Shreth Saxena, and Emily Wood. And then, of course, we have many, many volunteers. I hope I have everyone's name on here, but I might have missed a few people, but as you can imagine, it takes a lot of people to put together a conference like this. So what else? Oh yeah, Discover Live Lab. This will be circulating in between, this slide will be circulating in between talks, so you can always look at it there. And then I just wanted to also say a word about how we sort of organize this conference. It's a little bit unusual because we have 4 main speakers who give hour-long talks, and you'll see the schedule has lots of time for discussion, and interaction. And so this conference is really set up so that we can delve deeply into some topics and then we can discuss them. It's a small enough conference that we can have real discussions here in the formal setting, but also out in the halls and the other places. So please make use of that time to think and to interact. And finally, I just should acknowledge the sponsors of the conference. So with that introduction, we can get started, and Rafa is going to come up and introduce our first speaker.
Thank you all for coming another year to NeuroMusic. To me, it's a real pleasure to introduce the first of our four speakers, Dr. Sahar Moghimi. Listening to Sahar is always a scientific delight. She brings us new insight each year into the neonatal and prenatal brain, but for me, it's also a personal pleasure to introduce her. So Dr. Sahar Moghimi is a professor of neuroscience in the multimodal analysis of brain function research group at the Université de Picardie Jules Verne in Amiens, forgive my French. Before joining the group in 2020, she served as an associate professor of Ferdowsi University in Iran. In Amiens, Sahar team works closely, indeed within, the neonatal unit of the Amiens Picardie University Hospital Center, and this unique setting, working within the hospital, allows them to study the brains of newborns in an extraordinary and pioneering way, including the brains of preterm babies, and as you can guess, months before the typical 40-week mark of gestation. So here's my personal note. I met Sahar exactly 2 years ago, October 2023, although it feels like yesterday to me, during a turbulent transition between when I was finishing my PhD and my current postdoc here in McMaster. And at that moment, I was experiencing visa issues, and Sahar asked me to join the lab for a couple of weeks with them. And during that impasse of time, I see how they were working and her team and all the studies. And since the very, very beginning of cognitive life was deeply inspiring to me to see that experience and how they were studying. So as you listen to her, I invite you to reflect not only on the technical and scientific challenges she addressed, but also on the delicacy of the subject that they study, and also at the many families' stories behind the NICU walls that they involve in their study. So, as you might have guessed, her research focuses on early neurodevelopment, the link between spontaneous neural activity and the developing brain's first steps in processing exogenous information. And she used advanced neuroimaging and statistical techniques. And she's the principal investigator of several national and international projects examining the impact of premature birth and early musical intervention. So according to Scopus, that as you know is a little bit more modest in number than a scholar, she has published more than 50 papers and has collaborated with 92 researchers around the world. So in today's talk, Sahar will review recent findings on early neural capacities for auditory rhythm processing and how these capacities emerge and evolve from the beginning of the third semester of gestation to young infants at 18 months. And recently, I learned that Sahar loves jazz, and especially piano. And it's a pity she couldn't be here last weekend in our lab, in your jazz duo concert, but we'll have today our NeuroMusic concert. So this evening, I hope that you enjoy, and also she's gonna enjoy for sure our concert. So please join me in welcoming Dr. Sahar Moghimi.
So thank you, Rafa. Thank you very much for this introduction that touches me deeply. Thank you very much. So before starting, I would like to thank all the organizers and for this wonderful event, and especially Laurel for the invitation. It's very interesting for me, I mean, amazing, because the majority of what I will be presenting today is in fact carried out in collaboration with Laurel. So you can imagine how wonderful I'm feeling being at the University of McMaster and presenting these studies to you all. So thank you very much. So I will be presenting a project that has been going on in our lab for a few years now on the development of rhythm perception, auditory rhythm perception, the setup probably. And like Rafa mentioned, our population, that's probably one of the reasons that I'm speaking first, because the population that we are working on is the youngest. And so it's the setup, basically, we can imagine. And I'm coming from the University of Picardie, which is located in Amiens, a city in the north of France. And Amiens is known for its, so it's a small city, like 1.5 hours north of Paris. But Amiens is known for its cathedral. and also the ortillonnage, the floating gardens. And if you've been growing up reading Jules Verne, so Amiens is also the city of Jules Verne, so if you visit once Amiens, you can see his house and where he was working on his books during the last 34 years of his life, I think. So our laboratory is located at the University Hospital, which is like outside the city, and we are highly concentrated on the very early developmental stages. And it is very helpful because the lab is, in fact, the platform is located somewhere between the NICU, the maternity unit, the neuropediatry services, and so these all helps us to address our questions in a way that can be possible, in fact, otherwise it would have been very difficult. So we go to the NICU for most of our questions, and we record the neural activity using different techniques in premature newborns. So we bring the instrumentation to them and not bringing them to the lab. And we develop models basically based on the preterm period. So the premature neonates, the preterm neonates. And I think that here everybody knows this, but just because we will be talking about premature birth, preterm neonates. So we know that the normal birth is happening after 38, 40 weeks of pregnancy, and we consider neonates born before 37 weeks gestational age. So gestational age is counted as the first day of the last menstrual cycle of the mother, and we consider neonates born before 37 weeks gestational age as preterm newborns. And the degree of prematurity changes depending on the gestational age, from extremely preterm birth before 28 weeks gestational age, and then this goes on until 37. So we develop models based on these preterm newborns, and we use different techniques, depending, of course, on the question that we ask. We use high-density EEG, and before saying this, so in most of hospitals in France, but also in other countries now, preterm neonates get usual clinical routine EEGs from the date of their birth until the date that they leave the hospital to evaluate their cerebral maturation. So what we do, for example, is we go inside next to the clinical routines and where the neural activity is recorded, and we add protocols. So they get the EEG even in clinical routines without any research protocols. So we try to have our research protocols together with the clinical routines in these infants. So we use EEG. We use high-density and low-density near-infrared spectroscopy, and we recently started and developed OPM-MEG in the lab. Of course, for the OPM-MEG, the preterms and neonates have to come to the children, so we do this only near the age of term. We don't do it in very young preterms, and we are working to see if we can record the fetal activity, the neural activity in the fetus using fetal MEG, but that's a challenge, and this is still ongoing. So what you see here with the dashed rectangle, this is the period that recording is happening. So because preterm newborns starting from 24, 25 weeks gestational age, they're in the NICU. And this is the period, so between 24, 25 weeks gestational age to the age of term, where we use neuromonitoring to evaluate cerebral maturation. So, and if you look at the image, you see that during this period, but also after, there are different neurodevelopmental phenomena going on in parallel. But what is interesting is that, of course, starting before, but if you look at this particular window, we know basically based on animal studies that these phenomena are guided by genetic events, but also in very young, in very early developmental stages, based on the spontaneous endogenous neural activity of the brain. And then later on, also, but still before the age of term, by the information that arrives to the cortex from the external world. So there is a period, it's progressive, but first spontaneous activity, but also later on the external information that is guiding the neurodevelopment during this early stage. So if we consider particularly the auditory system, we know that the auditory system becomes functional really early, starting probably 24, 25 weeks gestational age. And if we consider connections in the brain, we know that the thalamic afferents that brings the information to the cortex in the young, very early period, so before 26, between 20 and 26 weeks gestational age, they arrive at the subplate, but the layer that is in blue, so which is just beneath the cortical plate, and this layer appears but then evolves and disappears near the age of term. But then at the beginning of the third trimester of gestation, probably around 27, 28 weeks gestational age, these afferents arrive at the cortical plate, so the cortex, and then later they develop synapses with the neurons in the layer 4 of the brain. And so the cortex becomes functionally sensory driven. So we can say that, okay, probably at the beginning of the third trimester of gestation, the cortex becomes connected to the outside world. So if we consider this window again, we have these two dimensions, the endogenous dimension and also the exogenous dimension. And what we think and what we try to do is that if we want to have a good evaluation of cerebral maturation during this period, we need to consider both dimensions, not only how the spontaneous activity evolves, but also how the brain starts to respond to exogenous information. And notably, in the auditory domain, we have shown recently that adding how the brain responds to simple auditory tones in terms of the spatio-temporal complexity of the response that the brain gives to some sort of small, simple tone stimulation. This adds, in fact, and improves the prediction of cerebral maturation, notably during the third trimester of gestation, insisting on the importance of considering the exogenous dimension into evaluation of cerebral maturation. But today, I want to talk about the project that we've been working on during the past few years on auditory rhythm, so specific characteristic of the auditory events. And I think, so what I will be doing is that I will show you the different studies that have been carrying out in our lab on auditory rhythm processing during this preterm period. And I think with all that I've seen today and all that we know from McMasters, I don't need to explain and convince you why rhythm processing is important. But we know that temporal and rhythmic regularities help different cognitive functions, and notably for language processing or processing of music, these rhythmic regularities allow chunking, grouping of events, and helping perception, and that synchronization to these rhythmic regularities help creating predictions about the upcoming events and the content, and also driving attention to certain moments of time. But from the developmental point of view, we know that the world of infants is very rhythmic, right? With all the infant-directed singing, infant-directed speech, the movement during the interactions between the caregiver and the infant, and also all that is happening. So the world of the infants growing up is very rhythmic, and that rhythmic regularities, time processing, and rhythm processing are cornerstones of many developmental and cognitive functions, including language development, musical behavior, social interactions, communications, and also recently based on studies carried out by different teams present here, that many developmental disorders are also associated with deficits in time and rhythm processing. And considering the neural data, we know that infants encode the temporal structure and rhythmic structure of sound in simple and syncopated rhythms. I think the first neural data comes from Laura Cyrilli's studies together with Laurel showing that exposed to a simple rhythm, the brain of the infants entrain or at least follow the rhythmic structure of the sound. But then Erica showed that infants can be primed to have rhythm perception on certain meters defined, depending on the priming. And also evidence is there for neural responses and also neural following of rhythmic structures in newborns. So this leads to the question, so how and when does this start? Why? Because I've just presented the development of the auditory system. So we know that the auditory system is functional before the age of term, so the capacities should be there. And also we know that the fetus is exposed to rhythmic regularities in the auditory domain, but also in other domains, for example, with the maternal rhythmic sounds, the heartbeat, the respiration, and also from different modalities, the walking pattern of the mother, and also this filtered version of the external sounds, either music or language that arrive to the fetus, passing through the maternal tissue, so probably filtered out for the high frequency content, but the low frequency content where rhythm is intact. So these information arrive to the fetus. So what are the capacities? What are the neural capacities? How do they set up during this period? So that's the question that we've been trying to address during the past few years. So I will show you a series of studies that have been carried out. So first, this was part of Mohammad Reza Adalati's thesis. At the time, he was my PhD student, and this was in collaboration. I think, if I forget to repeat it, everything is in collaboration with Laurel. And here we've been using the simple 6-bit rhythm that has been used in different neural studies from Laurel's team. So we recorded the EEG activity using high-density EEG in preterm newborns between 30 and 34 weeks gestational age. And we've seen that exposed to this simple rhythm, we see neural components emerging in the neural response to the rhythmic sequence, showing that the brain is following the rhythmic structure at different hierarchical levels. And this was not only for this rhythm, but also we've seen this in response to other stimuli, rhythmic stimuli, for example, the rhythm that we adapted from Sylvie Nozardon's studies, the 12-bit rhythm, and we see the same thing. We see neural responses to the rhythmic hierarchy. However, if we consider the neural response a bit further, we can see that, okay, if we have a series of evoked potentials, and we took a look at the spectrum of the response, then we can say that, okay, we see the response. So is this a series of evoked responses to a sequence of tones, or is there more to this neural response? Is this the rhythm, the regularity that is being encoded, or it's just the response to auditory stimulation? So to address this question further, we looked at the underlying oscillatory activity. And we saw that, so still we see the 6-bit rhythm that is created by a sequence of tone but rest events. And when we look at the oscillatory activity, so if you look at the rectangle marked over the time frequency representation of the neural response, we see these bursts of activity aligned to the beat of the event, of the rhythmic sequence, independent of the fact whether the beat is represented by a sound, by a tone, or a rest event. And the characteristics of these bursts of oscillatory activities is the same in terms of delay or power, whether it is sound or rest that is driving the beat. And this reminds of the studies in adults observing modulations in beta oscillations in adults for the perception of regularities and time perception, and leads to the hypothesis that probably it's not only a series of evoked responses to the sound, but there is more to it, and it is suggesting these predictive mechanisms involved in coding the temporal structure and rhythmic structure of the sequences. So there might be more to the response, and there might be, hopefully, what we think is that it is the temporal regularities, rhythmic regularities that is being encoded at this earliest stage of neurodevelopment. So having observed these results, we asked the question, okay, so we see these responses, but we know that the brain is going through fast development, structure, function, everything is changing, evolving. So how do these capacities develop? Is it something that we observe right away as soon as the brain, the cortex becomes connected through the thalamic afferent to the external world, or does it develop progressively? So we conducted a cross-sectional study covering the whole third trimester of gestation, starting from 27, 28 weeks gestational age until 38 weeks. And in this cross-sectional study, preterm newborns were recorded 1 week after their birth. So it's, we tried to sample the third trimester in a way to cover different gestational ages and so different developmental stages. And this was the study of Bahar Saadat Mer, my PhD student who graduated this year. And so Bahar developed a measure because extracting information from neonatal data is very, very difficult compared to adults. So we have to always work to find a solution. So we can see tendencies or even not, but we also always have to work on ways to extract the neural response. So we try to find solutions to see if we can have some information, we can extract some information or not. So yeah, so Bahar developed a measure because it was difficult to see the developmental trend to evaluate the neural response to the different periodicities present in the rhythmic structures. So what she did is that this is still the 6-bit rhythm that we are talking about. So the regularities at different periodicities, the underlying beat at 3.3, the grouping by 2, so the duple meter by 1.6, and also the triple meter grouping by 3. These are very periodic because it is the sound, we have a very regular sequence despite the fact that we have tone and rest events. So we decided to present the different periodicities by simple sinusoidals to have a solid representation of the periodicity, and then look at the narrowband EEG responses at the corresponding frequencies, so for the beat, for the grouping of the beat, et cetera, and to see how well these two signals, so the one representing the periodicity and the one corresponding to the neural response, they're aligned. So we get a complex measure, the strength of which gives the strength of coupling, how well they're aligned, and the phase represents the delay between the two. So in this study, we had 46 preterm newborns. And so what you see here, in the scatter plot, in the x-axis, you have the gestational age at the time of recording, 1 week after birth. And on the y-axis, you have the coupling strength. And what we see is that the response gets stronger. The infants get better with increasing gestational age at birth. The neural following gets better at all different levels. So the basic beat, but also the metrical hierarchies, the duple and the triple meters. So neural synchronization or neural following gets better with increasing gestational age at birth. However, when we divided the population in two subpopulations, those preterm newborns born before 33 weeks gestational age and those born after 33 weeks gestational age to compare. So 33 was chosen only because we wanted to divide the population in two. So we did this, and what you see here in black is the random distribution corresponding to the permutation of data, and we have the dashed vertical line. So we have two arrows, one in this yellowish color representing those preterms born before 33, and in orange or brownish, I don't know, those born after 33 weeks gestational age. So if you are on the right side of the vertical line, it means that the response is significant. If you're on the left side, it means that the neural following was not significant. So if we look at the fast periodicity at the beat, we see that both groups are to the right, so meaning that neural following is happening for this basic fast periodicity corresponding to the beat. But however, so this is for the fast periodicity representing the beat. However, if we look at the slower periodicities, so grouping of the beats by 2 and by 3, we see that the brain of older preterms near the age of term is doing it. So we have the neural response for the older preterms. However, the ones of the younger preterms, the response is not there yet. So neural following is not happening, suggesting probably that there is this maturational trend and the response evolves and gets better with increasing gestational age at birth. So what is the reason behind this? Is it the frequency, tempo, or cycle duration that makes it difficult for slower frequencies for the newborn's brain to follow the periodicity, or is it something related to the grouping of the events that is related to the metrical hierarchy? So to dig deeper, we conducted another study which is not published yet, so we're working on it. So this was a collaboration between Mohammad Reza, who now has a research engineering position at our lab, and with Maya from McMaster's, recording both infants and adults. So what we did is that we had 2 different versions of the same rhythm. The original version called slow tempo, the original tempo with the beat at 3.3 hertz, but then a faster version where we doubled the frequency. So the beat here is at 6.6, duple meter 3.3, and triple meter 2.2. And what we paid attention to was the cycle duration, meaning we kept the cycle duration the same, moving from the slow tempo to the fast tempo, so that the energy at the different metrical hierarchies would be comparable between the slow and the fast version. So we exposed with the same protocol, exactly the same protocol, newborns and adults to the same rhythm. And what you see here is the neural coupling normalized in the response in neonates and in adults to the same rhythm. So in 2 colors, we have in green the original tempo, and in orange the faster tempo of the same rhythmic structure. So it is interesting. The trend is, I think, very interesting. So if you compare neonates with adults, it seems like that in the neonatal brain, what counts most is the frequency or the tempo of the sequence of the periodicity, meaning that we get the best response for both tempi at 3.3 hertz. Just before going into the results, so response at every frequency is significant. I didn't go through the details. We are just comparing the patterns here. So we see that the best neural following is happening at 3.3 hertz at both the slow and fast tempi. So it's like it is the tempo or the frequency that is deciding the neural response, whereas when we look in adults, independent of the tempo, we get of the frequency, we get the best response at the duple meter, and it is interesting because the rhythm is a bit biased towards the duple meter. But so, for what matters more, it seems like for adults, is the metrical structure. So if we want to conclude, it's like in neonates, tempo plays an important role. However, in adults, the higher order metrical processing of the higher order metrical structure that is happening, arriving at much later developmental stages, is driving the neural response. So this is, of course, we have this interaction between the spontaneous development of the brain, because we know that the brain is developing, but also all that is related to the environment and learning from the environment that can play a role in shaping the neural response. So this is a question that we have to dig deeper. But for the neonatal period, for going back to our cross-sectional study, what we try to do is that, because if you look at the spontaneous neural activity during this period, I showed you, I just put some examples. What happens is that the spontaneous activity evolves very fast as well. So we go through this EEG in younger infants that is highly discontinuous. We have these bursts of neural activity separated by periods of silence or non-activity. And also we observe, if we look at the spectrum of the EEG responses, we see this emergence of faster oscillations in the spontaneous neural activity. So during this period, we ask the question, because we know that the neural interactions that shape these periodic, aperiodic characteristics of the spontaneous activity also can also play an important role in shaping how the brain, in fact, is responding to the exogenous information and exogenous stimulation. So what we try to do for now is to dig deeper. This is a collaboration with Mariam Horbani. We are developing a thalamocortical model that would mimic the spontaneous neural activity that we record at different gestational ages. And looking only at the spontaneous dimension, we want to see how the development of excitatory, inhibitory interactions and synapses, development of thalamocortical synapses, and the balance, the inhibitory excitatory balance can shape how the brain responds to stimulation arriving to the TC neurons in this thalamocortical model. So this is an ongoing study, but this does not consider, of course, the factor of the environment, which we know is playing an important role. So this is to come next, probably, and we will be having some results going out. So just to give you a teaser. But then, having observed this developmental trend and seeing that, okay, the response is there, but it's developing progressively, we ask the question, so what are the cortical structures involved in this processing? Because this is something that we could not address using EEG. So the question comes from the fact that based on the studies conducted in adults, we know that the motor system, and at the cortical level, notably the motor premotor regions of the brain, play an important role in temporal processing, in rhythm processing, and also the processing in general of temporal regularities. So we asked the question, so what are the underlying cortical structures for the response that we are observing? Is this something that is limited to the auditory cortex, or it's something that goes beyond, like what we observe in later developmental stages, something that goes beyond the auditory cortex into probably the motor, premotor areas of the brain? So to address this question, we conducted study using high-density near-infrared spectroscopy, again in preterm neonates, but here between 33 and 36 weeks gestational age, so a bit closer to the age of term than the previous study. And so we developed these patches because we wanted to have a high-density installation. We used fiber optics. But the problem is that so you can imagine the head size at this stage of neurodevelopment, right? So we had some difficulties holding the patches in place using the high-density montage and using the fiber optics. So the limit, I say the limit before presenting the studies, is that we couldn't cover more central regions of the brain. So we couldn't reach, for example, what might be an equivalent of, for example, SMA or more dorsal parts of the motor cortex. But anyway, we recorded high-density NIRS in preterm neonates, and we used rhythmic sequences adapted from studies conducted by Jessica Gran. And we had two conditions. We had the rhythmic condition with the rhythm highly regular and strong meter, and then a condition where we completely destroyed rhythmic regularities. So if we look at the spectrum of the stimuli, you see clear peaks corresponding to the periodicity present in the rhythmic condition. So just an example. And so, and this is an example of the rhythmic condition, and then we have the arrhythmic condition where rhythmicity is completely destroyed, and we have something as an example like this. So we see that there is the temporal regularities are destroyed. And we paid attention between the two conditions to have similar duration, similar intensity for the stimuli. So if we observe a difference between the two conditions, it's more related to the regularities rather than the acoustic characteristics of the stimuli. So when we look at the neural response, so in red we have the HBO curve, and in blue the deoxyhemoglobin. So in red oxyhemoglobin, and in blue the deoxyhemoglobin curves. And this is a typical hemodynamic response to stimulation with this positive polarity for the hemoglobin and negative for the deoxyhemoglobin. So we know that the brain is responding to the auditory stimulation, but what about the rhythmicity? So when we compare the two conditions, rhythmic versus arrhythmic, we see notably not over the auditory cortex when we don't see any differences between the two conditions. So at the bottom, between the dashed rectangle, we have the response, the ROI that corresponds to the auditory cortex, and in a continuous line, we have ROIs representing two probably motor and premotor cortices. I mean, what we have of these regions in our patches and sensor places. So what we see is that over these regions, we see an increased hemodynamic response to the rhythmic stimuli compared to arrhythmic stimuli, suggesting that the rhythmicity of the stimulus plays an important role and drives the cortical response in these regions, and suggesting that the encoding of these temporal regularities might involve regions beyond the auditory cortex and into premotor motor cortices of the brain. And we have hypotheses that needs to be addressed. But so these are only hypotheses because we know at this developmental stage, I'm not suggesting that we have the same thing at this early stage that we see in adults, but we know based on different studies that there might be this sort of underdevelopment coupling, long distance coupling at this early stage of development that might be involved in this wide distance networks and distributed networks involved in processing these exogenous and, in our case, these auditory rhythmic stimuli, even at this early stage. So having observed these results, what we try to do now, so this is the work of Monica Hedge, who is a postdoc researcher in my lab. We are trying to address, okay, so if these regions are involved, can we modulate in sort of a top-down way the neural response to rhythmicity, and can we have, because what we know is that somatosensory stimulation in neonates, notably, is this sort of very natural stimulation, right? Caregivers touch their babies with tapping, soothing tapping during, for example, infant-directed singing, infant-directed speech. So this is a very natural sort of stimulation, source of stimulation at this stage of neurodevelopment. So what we try to do is that to see whether this type of stimulation can modulate and bias rhythm perception in neonates. And what we try to do, but this is ongoing research, is that we simply tap, the experimenter tap the neonate in synchrony with the infant-directed song. So what the experimenter is hearing the metronome that you hear in this example. It's not working. And we have recorded infant-directed singing, and we have blocks.
Vous dirai-je, maman, ce qui cause mon tourment.
So what we aim to do. So what we try to do is to see whether this periodicity induced by this tapping can have impact compared to a condition when the tapping is not happening, so sort of top-down modulation of rhythm perception. So, but this is ongoing, so we have to see, we have some initial tendencies for the results, which are which are giving us hope, but we have to wait and see. And that's one of the problems in neonatal studies, is that we need a lot of subjects, participants, infants to get the response, like 30, 20, sometimes very difficult to get. You might see tendencies, but to have something more solid, we need larger populations. So, so this is the first part of my presentation. Before going to the next part, probably sort of small resume of what I just presented. So we've seen that so far that there is this developing neural architecture already in place during this preterm period for processing auditory rhythms. And this is, as we expect, something that is developing progressively, for example, with tempo or cycle duration playing an important role, with neural responses, neural following of slower periodicities arriving during later developmental stages compared to fast periodicities. And also that it seems likely that this early neural processing of auditory rhythms involve cortical regions, at least beyond the auditory cortex, and so distributed cortical regions involved in this processing. But you know what this means? It means that, okay, if these capacities exist already in the brain, and we already, I've talked about the impact of the exogenous information in guiding, in orchestrating neurodevelopmental phenomena, it means that, okay, these capacities are there, and also the brain is exposed to these information, all that is temporal regularities, rhythmic regularities. So they normally should be involved in guiding neurodevelopment and development of neural circuits, even at this early stage of neurodevelopment. So what happens is that if you consider the population that we are working on, the preterm neonates, they're deprived, depending on the gestational age at birth, they're deprived of this type of exposure. The rhythmicity, regular rhythmicities, is missing. More and more in the NICU, the medical doctors and medical teams are encouraging parents and caregivers to be involved in the neonatal life in the NICU, meaning with the congru time, being present, singing, speaking to the infants, and so providing stimulation. But if we compare to the natural development environment of the fetus, we can imagine that there is a deprivation of this sort of stimulation. So the question is, does this impact the early development during this stage, and probably does it have an impact in later cognitive development of this vulnerable population that corresponds to preterm newborns? But the problem with this study is that when we look, for example, at late cognitive measures or those carried out during infancy, is that, okay, there is a difference between full-term newborns and preterm newborns during this preterm period, but when they go back home, they're exposed to their home environment, which is highly variable from one family to another, and the amount of stimulation is not necessarily the same. So this complicates the evaluation that is being carried out during infancy and developmental measures to address the impact of preterm birth. So it's very complicated. So we need probably more rigorous measures that would take into account the home environment when they go home. So okay, what is the amount of stimulation? What type of stimulation? How much interaction? So probably if we can find a way, and there are studies that are developing techniques to better model the home environment with auditory environment or other types of interactions. So we need to integrate these measures in our models. But also what we try to do now is that, so what is the impact of preterm birth? So for example, those born really early at the neural capacities at the age of term before they leave the hospital. So that's what we should do, and we will be trying to do in the next steps of our project. But also another thing that has been going on during the past 2 years now in Amiens this is in collaboration with Laurel and also with Barbara Tillman. We are conducting musical interventions in the NICU. So starting from the beginning of the 3rd trimester of gestation, the infants born preterm are receiving interventions with live musical interventions, but also musical interventions with recorded music that were carried out in the studio with the help of our music therapist. And then we evaluate using mostly EEG, their neural response and development until the age of term. And then, so the website of the project, I put the address, so if you're interested. There are examples of the music stimuli that we are using now. And then we carry out follow-up studies at 6 months, at 18 months with these infants. So they come back to the lab, and we have different protocols using eye tracking, using EEG, and something that is very new for us, behavioral studies, where the infants tap, for example, in synchrony with the mother. And we are replicating and doing one of the protocols carried out here with Laura Cirelli with interpersonal synchrony with infants and prosocial behavior. So we have different protocols going on, and we try to look into the development of preterm newborns as a function of musical interventions. But still, again, this is an ongoing study that we have to wait a bit more to have more results. Okay, so for the last slides of my presentation, I would like to share with you an unpublished study that we started last year in the lab. So this is not neuro, this is something new. This is based on eye movement, so eye tracking of infants, so a bit later, exposed to infant-directed singing and infant-directed speech. So again, this is a collaboration with Laurel, but also with Rafa, with Gabrielle. I mean, who helped us in the development, and also Miriam Lenz for all her thoughts and suggestions and collaborations. So just give you a brief introduction. So if you consider adult studies conducted in adults, we know that the ocular movement, eye movement in adults, it seems like it's tracking the envelope, so amplitude modulation of speech, attended speech, so we need attention. And also what is interesting, lately found that it is even there in blind participants. So it's not something that is purely visual. It is something that probably related to the auditory modality, and that it's not only the amplitude modulation, but also the structure of attended speech. The structure of the sentence is followed by the ocular movement and also by blinking, but also the movement in general. Then we have these beautiful studies carried out in infants, notably by Miriam Lenz, who showed that in a natural type of stimulation, infant-directed singing, the caregiver adapts her behavior, facial gesture, to the strong beat of the song, and that exposed to these stimuli, the infant's eyes, so looking, comes back to the eyes of the caregiver on the strong beat, insisting on the importance of the strong beat and regularities during this type of interaction, and that there is a developmental trend and infants get better with increasing age at this behavior. So with these wonderful studies and observations, we asked the question, so do we have this sort of entrainment or ocular following of the rhythmic structure, and what happens beyond the strong beat? So can we see something similar to what we observe in adults, and what is the developmental trend? So this is the work of Betania Georlet, who is my PhD student, in co-direction with Barbara Tillman, and this is a study involved with infants involved between 6 and 18 months of age. So we probably know the background from Miriam's studies with these birds and everything, which is highly attractive for the infants, by the way. So thanks again, Miriam, for sharing all the information and all the stimuli. So this is a French version, but here you have, Rafa is conducting an English-Spanish version of the study, and we see similar results, which is very reassuring. So to give you an idea, so we have two conditions, infant-directed singing and infant-directed speech. So singing.
À la mode, savez-vous planter des choux? À la mode de chez nous.
So you see the instruction for the actresses is that you should imagine that you're doing this for an infant in a very natural way. So you see that the actress is moving, is doing all the things to as if something is happening naturally. And this is singing, but we have also speech condition.
Et qu'est-ce qu'il y a à la plage? Il y a le sable pour construire des châteaux de sable. Il y a la mer pour pouvoir se baigner et regarder tous les poissons de toutes les couleurs.
So this is the speech condition, and we have different stimuli, and the infants are, no, we already listened to it. So the infants are exposed to these stimuli, and we have a very clear, notably in the singing condition, auditory rhythmicity that we see in the envelope of the song, but also in the spectral flux, with the peaks probably better clear in the spectral flux. But interestingly, when we look at the spectrum of the spectral flux, so in black, we see these components corresponding to the periodicity, but when we look at the behavior, ocular movement, and here we use velocity of the gaze, we can discuss after why velocity and why not the trajectory of eye movement, but I think we can leave this for further discussions. So we see similar peaks. But what is important is that because this is sort of natural stimulation for the actresses, rhythmicity in the auditory domain is highly associated with rhythmicity in the visual domain as well, because there, as an example that I just showed you, see the singer that is moving on the beat notably, and we see that if you consider the movement of the singer's head, we see peaks also corresponding to the beat in the movement, but also all these gestures that Miriam also observed in her studies with, for example, singer opening the eyes on the strong beat. So we have all these visual information related to the rhythmicity, but also the auditory domain. So we thought, okay, how can we model the role of the auditory rhythmicity driving the infant's eye movement? So we developed a model based on linear kernel-based analysis, regression models, and in the first scenario, we only input to this model the visual information related to singer's head movement and also, for example, wide eyes on the beat. And we try to predict the eye movement behavior as an output, and we compare this to the actual signal. Then, in the second scenario, we feed in also the spectral flux, so the auditory rhythmicity as a second input, and we try to see whether the prediction of the actual signal improves as a function of this second input. And then also we compare this with the random condition. So we have, because we know in these regression models, adding an input can improve predictions, but without predictive value. So we need to make comparisons with random conditions, showing that it is the regularity and not just increasing the dimension that is driving the response. So I will show you some results of this study. So first, for infant-directed singing. So in green, you have the response to the first model, only the visual dimension, and in blue, the second scenario where we have both the auditory and visual results. And we have a very strict permutation case, which makes the results probably not, the difference not very visible, but anyway, we see this in both conditions. So the distribution corresponds to the random condition. Again, if you're on the right of the vertical line, it means that the difference is following is significant. On the left, it's not the case. So we see that for both models, the first visual model, but also the second model, audiovisual model, following is happening and we see how we have a good prediction of the eye movement behavior. But what is interesting is on the left, is that there is a significant difference between the two models, suggesting that adding the auditory rhythmicity improves the prediction about the eye movement of the infant. So auditory input is playing an important role in predicting the eye movement behavior. However, when we look at the speech condition, so this is exactly the same scenario, it's just that the input is different. We are looking at the speech videos. We see that for both conditions, both auditory and visual, the response is marginally there. So ocular following is happening, probably, we don't know. So it's not the observation that we have in adults where the ocular following is happening. It's not there yet or marginally there. But what is important is that adding the auditory input does not improve the prediction. So what is the reason? So we know that this is observed in adults. So we have different hypotheses about this. So it might be the fact that the rhythmic regularity is less present, or speech is less regular compared to the singing condition, and also the fact that the infants that we included in our study were between 6 and 18 months. So probably this can improve with development of language, and probably we need to extend our study into later developmental stages, at least to see what is the trend. And if they arrive to do this, we get this ocular movement and ocular following of the rhythmicity in later developmental stages. So this is the next steps that we have to go through to dig deeper in this new line of research that we are developing in the lab. So with this, I finish my presentation. I would like to acknowledge my team, my PhD students, and my and postdoc researchers that are working with me. Also, all the team members from the University Hospital, Pauline, Brunel, and also medical doctors from the NICU, and our music therapist, Stephanie Lefebvre, and also all the collaborators of this project. I didn't mention all of them, but especially Laurel Trainer, Barbara Tillman, and last but not least, definitely Fabrice Valbois, without whom none of this would have been possible, and also the founding agencies. And at last, thank you very much for listening.
Now we have time for questions. There. They're bringing the mic.
Thank you. Hello, hello, hello. Okay, cool, sorry. Yeah, this was so nice to see some of these new findings, and I actually have two questions. The first, since it's more recent, I guess we can go with that. With the last study that you're talking about with the eye tracking and the visual tracking, was there any plan to do an auditory-only model to see if you still get significant prediction if you ignore the visual information?
So that's a very good question. Yeah, so that would be the next step, but I know that Rafa has been doing that here with trying to separate auditory visual dimensions. It's a bit complicated because we cannot really conduct a fixation task in infants, so we need to have something that would attract their attention to the model. But one of the solutions is something, for example, Rafa is doing similar to this or in other ways to try to separate the visual and auditory dimensions by associating, for example, visual stimuli that are not really related to the auditory rhythmicity and creating different conditions, for example, keeping the visual but making the auditory random, or vice versa, trying to address this question. But what I forgot to mention is that what is interesting, I mean, and that's one of the next steps that we're taking, is to add the neural data and trying to extract also the ocular components in the neural data and to look, because what is the, I'm benefiting from your question, right? So the question is, so what is the reason? Is the ocular movement helping some function in the brain, or is it the consequence of something that is happening in the brain, right? So this is the question. So do we have some more like global entrainment that is happening over these oculomotor areas, or something that is related into attention? So yeah, we need to address all these questions, but one of the important items that need to be addressed is the separation of the auditory visual components, right?
Sounds like not an easy task, but yeah, super cool. And then my other question was about the rhythmic tapping study. So I know that you didn't go super into it, but I was just wondering, so the mom is tapping and singing, and then she stops for a while, and you're comparing those two conditions. So I guess I'm just wondering, are you planning to make sure that she's not also modulating the amplitude of her singing when she's tapping? Because I could imagine that, like, if you're tapping, you might also modulate your auditory information, so it might not be like the most perfect comparison, I guess. And yeah, just wondering if you've thought of that.
I didn't go into the details, so that I should have probably. So we're using recorded songs. So it's our music therapist's voice recorded, and which is being played for all the infants. And it's not the mother tapping; it's the experimenter. So it's the same for all the babies. And we have two conditions. Once it's only the song, so the metronome is not played for the baby. It's in the experimenter's headphone, trying to keep the beat. And we have two conditions: no tapping at all, and a separate condition where she's tapping only on 4 beats. So ta, 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 and then waiting for 4 beats and doing it again. And we're comparing these periods where no tapping is happening between the two conditions. So auditory stimulus is the same, and only the impact of tapping is changing. And we see, because tapping is happening at 1 hertz, and we see this, so it's sort of a teaser, but we see this tendency for the impact, at least we only have 25 neurons at the moment. We see these tendencies for improved components at 1 hertz, but that's to come.
Thank you so much.
Thank you for the speech. Yes, thank you for the speech. I might have some stupid question. So what's the sound environment in NICU, especially when the preterm neonates, they live in the incubators? Are that incubators soundproof? So our preterm neonates live in more like regular sounds, like the machines in NICUs, rather than a normal term neonates. And for the mothers who sing and taps, I suppose they are not musicians, they are not percussionists. So they might produce more like AC than the RC situation. How do you think about it?
Yeah, so I think there are studies showing that it doesn't matter. I mean, there is no bad singer for at least at this period for the mother-infant interaction. It's the interaction that counts for, I mean, we don't look for highly regular behavior, but I see your point, what you're suggesting. So, but I'm not sure if I got the first question quite correctly. So you're saying that whether because of the alarms, which are regular, do we have more regular sounds in NICU for the that's your question? Okay, so yeah, so the alarms are there, but there are studies that address the impact of exposure to the alarms, suggesting that these infants are more sensitive later on to these sounds, for example, related to the timbre of the alarms. But I'm not sure that, but it's not always regular. They're on and off depending on the state of the. And alarms sometimes are only signaling the nurse outside and not always in sounds. It depends on the state of and how the regulation of the NICU, is happening do they need these alarms signaling for each infant? So the sound is present, not present, sound present, not present. But we didn't consider this factor in our models. It's just that when we're realizing the interventions, we turn off the alarms because there is the nurse and the technician present in the room. And this is the same between the preterms receiving and not receiving the interventions. But we haven't control for that. And yeah.
Hi, thanks for your talk. I was wondering if I could get you to speculate a little bit about the enhanced neural following that you saw just for those 3.33 hertz frequencies.
I didn't get the first part.
If I remember correctly, when you did showed your neural following responses for different like beat and duple and right, and it was enhanced for the neonates for 3.33 for the fast. Is that correct? Could you speculate a little bit about what you think might be happening? Why?
Why 3 hertz is better for both tempi, whether it's slow or fast? Yeah. So this is the case. So we see that it's like the frequency is more important than the metrical structure. So because when we go to the faster tempo, 6.6 might it seems like it's too fast, but we get better responses at 3.3 compared to 2.2. So we can have different hypothesis for that, because even in later developmental stages, we see that even in behavior, infants, young children and infants are faster in tapping or in behavior, and then they slow down, right, in the adults. Some of it, I mean, if we consider this, so there are different factors that can play a role. I mean, developmental-wise, it can, for example, if you even like in a very mechanistic model, if you play with the balance between inhibition and excitation, so you have synapses and you play with the inhibition-excitation balance, you can modify how the network, so like a neural mass model, nothing related directly to the brain, how the network would respond and entrain to sort of periodic stimulation. So if we try to extrapolate this to the infants, we might say that to some extent, the development that leads to this change in balance can have an impact in how the brain responds to different frequencies, but also with all that is structural, with the development between the auditory and the motor cortex, and the impact of the motor cortex that can change with the development of distributed networks and connections, can change, also impact, and also motor development by itself can impact how the brain responds to slower frequencies. I mean, and I'm not considering, and I don't discuss all that is environmental, right? So I think it's not one reason, one factor, but a mixture of all of these that are playing a role in changing how, and also learning, because in adults we see that, okay, we go to this slower frequency range, but the better response at metrical hierarchies, so all that is learning also can impact the shaping of the neural response. I don't, yeah.
Well, thanks for the talk. Do you hear me? Okay. Well, thank you. I have a question about the one of the few first slides that you shown the spectrogram and you compared with Fujioka paper. I'm not sure if you can get back to that slide right now, but if it's possible, it would be easier. What caught my attention was that this is what you observed was in the alpha band rhythm. It's 7 to 12, right? And what we've seen in adults, it's something around beta and gamma. And this is so interesting. So first of all, is there any filtering type of thing happen that we didn't observe beta and gamma activity, or?
That's a very good question, and that I forgot to address when I was
Yeah.
on this slide. So thank you for the question and bringing this
Sure.
back. So again, there might be different reasons, but one of our hypotheses, because you see, when we look at the when you look at the spontaneous activity, when we go to younger preterms, we don't have even spontaneously fast oscillations. So for example, when you look at 26, 27, 28, you really don't see beta in the spectrum. And then with increasing gestational age, even spontaneously, you have these faster oscillations arriving orchestrated by the slow oscillations, as we see, for example, sort of what we see in spindle slow oscillation synchronization in adults. So we see these sigma oscillations arriving around 28, 29 weeks gestational age. So the brain itself is not doing these fast oscillations at early developmental stages. And again, it is related to, we have different people working in animal models show different reasons for this, including the progressive development of inhibition in the brain, and this progressive but turning between the GABA, which is only excitatory, which is going more into inhibitory, so the interactions that lead to faster oscillations. So the oscillatory regimes underneath the neural response is not necessarily the same during its early stages compared to later stages in adults. So I think this is one of the reasons. But we need to pay attention that we shouldn't say that, I wouldn't say that this is alpha, equivalent of alpha, what we see in adults in time perception, because I think the functional role of alpha is not the same in early stages of neurodevelopment compared to later stages. So it is alpha frequency, but I wouldn't associate this with the alpha we see in time perception. But one of the reasons might be this, that we are in a very early stage of development and we don't see beta, gamma, and we are seeing probably slower frequencies in response for the response.
Yeah, so we could confirm it's type of network properties that develop later on and then they emerge. And that's what you mentioned is due to inhibitory ones mostly, right?
And the balance. So
and
because we have the development of
inhibitors.
this is one of the reasons. I'm sure that it's not the only one.
At least.
Yeah.
And what I can see here is that these emerging clusters judge right on just before the onset, am I right? So something similar to what we see in adults in a different region, right?
That's one of the...
so before initiation of the movement, right at the onset. Okay.
That's why we suggested that it might be something related, like predictive mechanisms related to the prediction of the time of the next beat, because it's preceding. It's not proceeding, it's preceding. But what is... so what we should have done, doing it again, is that we should have added because in adults we see that, for example, the beta modulation preceding can also impact how the brain responds to novelty, for example, like in terms of frequency changes on the sound, and the beta characteristics also predict how the brain would respond to novelty arriving at the time of the expected event. So we didn't have this condition in our stimuli, and it's difficult because mismatch responses are by itself are difficult to analyze. We have it, we see it, but like looking at the individual levels as trying to associate it with oscillatory activities, it's very complicated. But in an ideal scenario, we should have added this, and to see if the characteristics of these oscillatory modulation preceding the beat would predict how the brain would respond, for example, to what's happening on the beat. But that's probably next steps that we should address. Thank you.
Thank you so much for the talk. I was really intrigued by the finding that it's engaging really early on the premotor and motor cortex. So I'm curious, I guess it's a bit of a chicken and egg question, and how much they can move already right at these early stages. So is it that like the more they can move, it kind of rapidly increases, or Is it that like there is more neural development first and then they can move? So I'm just curious about this.
Moving like the baby by itself, you mean? So the very young preterms don't move a lot,
Right.
right?
Yeah.
But we have spontaneous. So it depends how you define movement, right? Because you can see like contractions, is it movement? Spontaneous contractions, is it really movement? Or movement is more like they have to move the limbs, they have to move the arms, etc. So they don't move a lot. I mean, they don't move when they're young. But when they get near the age of term, in the clinical evaluations, one of the factors that are being analyzed is spontaneous movement. So how they move and how much they move is one of the measures that is used for evaluating cerebral maturation of these infants. But in general, we don't observe. You have, because at this stage of neurodevelopment, you have spontaneous activities in the sensory systems, visual, auditory, like in the spontaneous activity in the cochlea or the visual retina, and also muscles that are expected. The hypothesis, which is shown in conferred in animal models, is that they play a role to develop the cortex. So the connection, you get, for example, twitches, and then you have activity in the motor or sensory cortices. So this is related to the developing connections. And these are completely spontaneous, but we don't have periodic movements. It's much, much, much later that they arrive.
Hi, great presentation, thank you. So in my practice, I utilize a lot of multi-lobe activation to build neural connectivity in the brain, mainly in kiddos 5 and up, into teenage years, autistic kiddos, ADHD kiddos. Now, I utilize a lot of brainwave entrainment using isochronic tones, binaural beats. What I use is vibrations, frequencies, so 20 hertz, 15 hertz, 10 hertz, 5 hertz. I'm curious if you prefer that or in addition to notes, so C, B, D, whatever you guys use, or is it just vibration that you find more effective?
Vibration related to the stimulation, you mean?
Mm-hmm. So yeah, if I'm training, say, focus or attention, I'm using 20 hertz. If I'm training relaxation, meditation, it's 10 hertz, 5 hertz, 15 hertz, things like that. But I'm really curious if I should add in tone, so like a C note or a D note or an E note or F note, while modulating that vibration.
So I'm not sure if I got your question correctly, but so the idea is that adding a different modality would improve the perception of the regularity. I think I think it's reasonable to assume that it would be the case even in early stages of development, because so you have these inputs of different modalities, and it's at least the somatosensory network that are first of all, we know that the response to like when you again. One of the things that are carried out in clinic is that to evaluate cerebral maturation, one of the things that they do is that they do somatosensory stimulation, that they look at the evoked potential, the characteristics of the evoked potential. So the system is already there. And we can imagine that timing is also important in both modalities. So my guess is that, and that's what we are doing with the tapping protocol, because we assume that this can have an impact. And so I don't remember, I was discussing this with, I think, Trin yesterday, and she was saying that, okay, you're doing it in newborns, but you should do it also at 2 months old when they have the development goes forward. So my guess, and that's one of the reasons we started this study, is that it should be there, this top-down modulation, and the type of stimulation is already there, and networks are developing. This is a premature version, but the connections seem to be there. So my guess is it should impact the multimodal stimulation. You can have this cross-modality impact from one modality to the other. But the idea is that how it develops and how much you get it at the beginning and how much you can get it later on.
And retention.
And?
And retention.
Yeah, exactly.
Do you find any tones work more than other ones? So if you're using different notes, any ones are more receptive at your population?
At this stage?
Yeah.
So, well, it's different. So, I mean, auditory-wise, this is the discussion that we were having for creating stimuli. If you want to get a neural response, probably it's one of the good solutions is to have wider band spectrum, because you stimulate more, a larger population of neurons, at least over the auditory cortex, right? Because single frequencies, smaller population, wide band spectra, larger populations. So, but then how much is this comfortable for them?
Yeah.
That's another question that have to be addressed. Yeah.
Okay. Great, thank you.
Thank you.
So I think we ran out of time, but we can still talk more during the lunch and on the conference. So please, let's thank again Sahar for